Welcome back, Bio 47. Today I'm going to finish up with the lecture material about Chapter 9, about the muscular system. You're going to want to make sure that you watch this, take notes, pay attention, because Quiz 5 is going to continue to, as it's regularly scheduled, on Monday. You're going to be able to take that on Canvas, so make sure that you review aspects of the muscular system related to everything that we've been talking about in lecture, in preparation for that quiz number 5. So we've been talking about the muscular system, which is this really highly modular system, meaning it's made up of these long subunits that get bundled together in regular patterns. So we have muscles that are made up of fascicles, that are made up of muscle fibers, that is the cells. Inside of the cells are myofibrils, those cylindrical organelles that are made up of myofilaments, individual strands of proteins, of the protein actin and myosin primarily. And all of these taking these really long cylindrical shapes that allow them to perform their function of contraction. So I'm not going to talk about the subcellular structures today, but if this looks unfamiliar to you, you might have a bad time when you try to take quiz number 5. So make sure that you go back and you review some of these subcellular structures of muscle fibers. In particular, the parts of muscle fibers have special names because they're highly modified to the jobs that they do. So the cell plasma membrane is no longer called the plasma membrane. It's called the sarcolemma. And those infoldings of the sarcolemma that bring the outside world in really close contact with the inside of the cell, those are those transverse tubules, or T-tubules. Inside of the cell is called the sarcoplasm, and it's completely filled with these myofibrils, these bundles of protein filaments. And in the sarcoplasm, you're going to find a lot of mitochondria. You're going to find the nuclei, shown here in blue, pushed to the sides. And lastly, the sarcoplasmic reticulum, that internal membrane complex that in other cells is responsible for lipid synthesis primarily, here is going to be responsible for the storage and release of calcium that allows for fiber contraction. So it's called the sarcoplasmic reticulum, and those enlarged areas are called the cisternae. They're in really close contact with the transverse tubules, and together that structure of 3 is called a triad. Another structure that you might want to be familiar with before you go to take a quiz is the structure of a sarcomere. So the sarcomeres are these structural and functional units within a myofibril. One sarcomere is considered the distance from one Z line or Z disc to the next Z line or Z disc. So it contains overlapping thick and thin filaments. Go back and review this section in your textbook and in my lecture slides if it's something that's looking unfamiliar to you or if you've missed some lectures recently. One more thing to take a look at is the anatomy of the neuromuscular junction. And in lecture on Monday, we went over the neuromuscular junction. We said it's a type of synapse, and that a synapse is any place where a neuron is communicating with some other cell. So a neuron is communicating with another neuron, or in this case, with a muscle fiber. So when a neuron is communicating with a muscle fiber, then it's called a neuromuscular junction, a type of synapse. So the presynaptic cell will always be a neuron, and it's sending messages across the synaptic cleft, a tiny open space between the neuron and the muscle fiber. So there's no actual direct contact. It's sending this neurotransmitter across a physical open space called the synaptic cleft. And I don't want to spend more time on this today either. Take a look back at this section of the book if you're feeling shaky about it or if you missed lecture. The last thing that we covered was the concept of a motor unit. So here's a zoomed out view of that neuromuscular junction, the NMJ. showing that there are muscle fibers, and each one of them is controlled by a single neuron, but that a single neuron can control multiple muscle fibers. So that's what's shown on the left in a cartoon version, and on the right, an electron microscope version. It's showing that a motor neuron is controlling multiple muscle fibers. So when a motor unit gets stimulated, when a message gets sent by that neuron, all of the muscle fibers to which that neuron is communicating contract at once. We also said that the size of the motor unit and the degree of control are inversely related. So really small motor units, like the ones shown here, where a neuron is controlling just 4 muscle fibers, that's going to provide really precise control. That would be things like in the eye muscles. For a larger motor unit, you might have a neuron controlling tens or hundreds of muscle fibers. That's going to be something like you'd find in the lower limb muscles. If you're trying to do a movement that requires more force, you would recruit more motor units. That is, you'd have more neurons firing. So now we're getting into a bit of new material, and this is the blood supply to muscles. Recall that muscles have a lot of mitochondria. They're doing a lot of activity, so they require a lot of oxygen to be carried to them via the blood supply, and they require a lot of carbon dioxide to be carried away. So they need a lot of blood supply. So take a minute, pause the video, because this is a good place to think about the modularity of muscle organization and see if you could fill in those 3 blanks. So hopefully you took a moment and actually saw that you could fill in those 3 blanks. So the first one, the big one, indicates an entire muscle. So that would be an entire muscle that's surrounded by epimysium. The slightly smaller one would be a fascicle, a bundle of cells that would be surrounded by perimysium. And then the last would be an individual muscle fiber or cell that's surrounded by endomysium. And throughout these connective tissue sheaths that surround the muscle, then we're going to be finding blood vessels that are providing blood to the exterior surface of the muscle and in between the fascicles. So there's blood supply that's traveling through those connective tissue sheaths, through the epimysium, through the perimysium. So I'm now going to talk for a bit about how muscles work and some differences that are possible between muscles and muscle fibers. So muscle tone refers to this constant tension that occurs within a resting muscle, and that seems sort of like a contradiction, right? If a muscle is resting, then it shouldn't be constantly in tension. But this is this continuous and passive partial contraction of the muscles, and it's that muscle's resistance to passive stretch during the resting state. It helps to maintain posture, and muscle tone declines during REM sleep. If there's a sudden pull or a stretch, then the body is going to respond by automatically increasing the muscle's tension. So this is what helps you maintain balance. If you're standing up, you're using muscle tone to maintain posture. And through the muscles that are helping you to stand up, motor units, those fibers that are stimulated by different neurons, they're stimulated randomly. So they're stimulated at different times to help avoid fatigue. It's not just one motor unit that's constantly working. So there are a couple of different types of muscle contraction that can result. In isometric contraction, the length of the muscle is constant, but the tension is changing. So in isometric contraction, the length of the muscle doesn't change because the tension never exceeds the resistance. So an example of isometric contraction would be if you tried to pick up a car, you could exert a lot of force. You could change the tension that you were exerting, but your muscle wouldn't get any shorter because the car wouldn't be moving. In isotonic contraction, this is where the tension is constant, but the length is actually changing. So this is where tension is exceeding resistance. For instance, if you're picking up a bag of groceries, the amount of force that you're putting forward is going to be the same because the weight of the bag of groceries stays the same, but you're going to be moving bones closer to each other, so the length is changing. Further, there are two types of isotonic contraction. The first one is concentric contraction, in which the muscle is shortening. This would be you picking a baby up from a crib, let's say. And eccentric contraction is when the muscle is lengthening, and that's when you're lowering an object back down, putting that baby back in the crib. This is where muscles are active while they're lengthening. So here's an illustration that shows both isometric and isotonic contraction. On the left is isometric contraction, and in this one, Nana is trying to curl that 80-pound bar, and she hasn't quite worked out enough yet to be able to lift that 80 pounds. I think she'll get there though. So she's showing us isometric contraction. The tension that she's able to exert is less than the resistance. So she keeps putting in force, but no movement is occurring. On the right, she can curl that 10 pounds with ease. So this is isotonic contraction. This is where the muscle tension is greater than the resistance, so we're getting movement. When she's curling up, when she's flexing her forearm at the elbow, that's concentric contraction. When she's extending her arm, when she's lowering the weight, that would be eccentric contraction. So those are movements that are produced by muscles. Within muscles, we're going to find different types of muscle fibers, so different types of cells. And essentially, there are two types of muscle fibers, and these muscle fibers vary in their contraction speed. So there's slow twitch muscle fibers and fast twitch muscle fibers. So the slow twitch muscle fibers, shown there on the left and in the darker color in this cross section of a muscle, these are slower, they're less powerful, they operate over long periods without fatigue, and they're fueled by oxygen, so they're aerobic. So they have steady power, they have endurance. The fast twitch muscle fibers, shown in the lighter color in the cross section, those are more numerous, they contract in short bursts, and they can make ATP even in the absence of oxygen. So they have these explosive powers, but they can fatigue easily. So fast twitch are those that are lighter, and slow oxidative, slow aerobic, those are the darker ones. So the slow twitch are fatigue resistant, and the fast twitch are more easily fatigued. So on the left, maybe there's a marathon runner. She's really got endurance because of this high concentration of slow twitch fibers. And on the right, maybe this is a power lifter, somebody who has explosive power, but will only do a couple of sets. And the skeletal muscles of an average person have about fast twitch, or about half fast twitch and about half slow twitch fibers. Among elite athletes like Olympians, proportions can really vary. So sprinters have about 80% of those light, fast twitch fibers, whereas marathoners have about 80% slow twitch fibers. And this is something that you might understand if you go home for Thanksgiving and your family cooks a turkey, and they might ask you, do you want the light meat or the dark meat? And what they're really asking is, do you want to eat a muscle of a bird that is primarily fast glycolytic, that fast twitch fiber, or do you want to eat a part of this bird that is primarily those slow twitch fibers? So those fast twitch fibers are the light meat And those slow twitch fibers are the dark meat. Remember that fast twitch are those that are more numerous, they contract in short bursts, and slow twitch are those that are slower, less powerful, they operate over long periods without fatigue. So where in a turkey do you find light or dark meat? In the turkey, you find thighs and drumsticks that are dark meat. This is because turkeys use their legs continuously. So the leg meat has these smaller diameter, slow twitch muscle fibers that have great resistance to fatigue. Domestic turkeys are so large that they don't use their chest muscles to fly very much. So their well-rested breasts become our white meat. And these have these fast glycolytic fibers. This would be something that they would pump really quickly to be able to fly, but our turkeys don't really fly very much. So the breast is the white meat, those explosive force fibers, whereas the legs are the dark meat, the ones that are continuously carrying the turkeys around. Let's get back to humans. So in humans, we find that a skeletal muscle usually contains both fiber types, that is the slow twitch and the fast twitch. However, a single motor unit, the fibers that are controlled by a single neuron, contains only muscle fibers of the same type. So a motor unit would either be slow twitch or fast twitch fibers. The slow twitch fibers dominate those postural muscles, like those in the back and in the calf, that are continuously working to hold you upright. So you don't want those to get fatigued because you do need to stand up all day. For muscles that require really swift but brief contractions, like those in the eye and in the hand, that's where you're going to find exclusively fast twitch fibers. I said that the proportion in Olympians can vary, and the relative proportion can really dictate what you're good at. For instance, a distance runner like myself probably has a higher proportion of those slow twitch oxidative fibers in my legs. I'm horrible at sprinting, so I probably have a really low proportion of those fast twitch fibers. The proportion that a person has is determined by genetics. So you're born with a particular proportion of slow twitch and fast twitch fibers, and you can't change that proportion that you have. However, your abilities can definitely be changed by conditioning. I could get better at running sprints if I were to increase my metabolic capacity, increase my conditioning. I just don't really want to. So another aspect of muscle function is the control of contractile force, which you have. So let's say that you went to go and pick up a paper coffee cup. You have to have really precise contractile force to be able to successfully pick up that coffee cup. If you tried to pick it up and you didn't exert enough contractile force, it would slip through your hand and fall on the ground. You'd lose all your coffee. If you exerted too much contractile force, you'd crush the cup in your hand like the Hulk, and you still have coffee all over yourself. So the contraction of muscles and control of contractile force is what allows us to perform everyday tasks. And the force that are generated by those contractions really has to be controlled. And it's controlled in two different ways. The first way is by controlling the frequency at which individual muscle fibers are stimulated. So if the frequency of stimulation from a motor neuron increases, then muscle tension in the motor unit within those individual muscle fibers continues to rise until it reaches a peak point. And that's when those muscle fibers are just contracting continually. So if the muscle fibers are getting a message from a neuron to contract, then they contract continually. The second way that contractile force can be controlled is by controlling how many muscle fibers take part. That is, how many motor units are being recruited. So if additional motor units are recruited, then there's going to be an increase in contractile strength within the muscle because more individual cells are contracting. Something that might be of interest to you all is figuring out how is it that you make your muscles bigger and what is it that's happening when muscles get bigger with exercise. So with exercise, you'll experience muscle hypertrophy. This is an increase in the fast muscle fiber size. What's actually happening when you build muscle? You're increasing the size of the fibers, that is the size of individual cells, but not the number of cells. So the number of muscle cells or muscle fibers within a given muscle is pretty much set throughout your life. It's the individual cells that get bigger when muscles get bigger or get smaller when muscles get smaller. How does this happen? The number of myofibrils per fiber increases. Essentially, you're packing more protein inside of the sarcolemma. So the sarcolemma is expanding, getting bigger. The individual cell is getting bigger. That means the individual muscle is getting bigger. Additionally, more mitochondria and more glycogen are stored in the cells. More water is going to be stored in the cells as a result. So the entire cell is getting bigger. That's what's driving the muscle getting bigger. And this growth in muscle fibers results from repetitive, exhaustive stimulation of the muscle. If you don't use it, you lose it in the case of muscles. This is muscle atrophy, and this is a wasting of muscle that reduces the fiber size. Again, you're not losing cells. Cells aren't dying, but the individual cells are breaking down myofibrils. They're recycling those proteins because they aren't being used. So if you don't use muscle, you're going to lose muscle size, you're going to lose myofibrils, and you're going to lose power. So let's talk about some of these actions of skeletal muscles, which you all probably know well from completing your homework number 3. So for the actions of skeletal muscles, the main mover of a particular action is said to be the agonist. So the agonist of a movement is the muscle whose contraction produces the movement. This is also called the prime mover. So the triceps brachii, that movement on the muscle on the back of the brachial region, that's the agonist for forearm extension at the elbow, or the prime mover of forearm extension. The antagonist is a muscle whose action opposes that of the agonist. So the biceps brachii would be the antagonist for forearm extension. It's said to be antagonistic to the triceps brachii. A synergist is any muscle that assists the agonist in performing its action. And these could be muscles like those that stabilize a joint. So for instance, if you're using your biceps brachii, if you're performing flexion of the forearm at the elbow, some muscles that are synergists would be those of the rotator cuff, those that are holding the head of the humerus in place in relation to the glenoid fossa and allowing for stabilization of the humerus. So the last thing that I'm going to discuss are the characteristics of these other 2 types of muscle. We talked about skeletal muscle. We're also going to talk about cardiac muscle and smooth muscle. And in contrast to skeletal muscle, which is voluntary, it's under your conscious control, these 2 types, cardiac muscle and smooth muscle, are involuntary. So the first type is cardiac muscle. These cardiac muscle cells are found only in the wall of the heart. And we'll spend a lot more time thinking about how the heart works when we get to the unit on the cardiovascular system. So it's this cardiac muscle, though, it's only found in the heart. It is striated, so it has that regular arrangement of myofilaments within the myofibrils. These cells, you're going to see 1 or 2 nuclei That's in contrast to skeletal muscle, where there are multiple nuclei per cell. And like skeletal muscle, there are going to be many mitochondria inside of these cells. The cells are branched. They're forming these Y-shaped branches that form multiple attachments to other cardiac muscle cells. So there's going to be a lot of interconnectedness between cardiac muscle cells. Another feature that allows for this interconnectedness are these intercalated discs that join adjacent cardiac muscle cells. So these structures attach adjacent cells. When the muscle contracts, cells would otherwise pull away from each other, but the intercalated discs hold them together. And they're made up of desmosomes, which we said are those cellular junctions that function like spot welds, proteins that knit plasma membranes together with gap junctions. And gap junctions are what allow the heart to function simultaneously across all the cells by sending messages between the cytosol of adjacent cells. So due to the branching and to the gap junctions, these muscle cells can function as a single unit. So they're said to be autorhythmic. They're able to generate electrical impulses without any nerve stimulation, and that nerve impulse just travels around the heart repeatedly due to the branching and the gap junctions. So they can function as a single unit. So it's under involuntary control that the heart continues to beat, although when it's still in the body, it's under the control of the autonomic or the unconscious nervous system, which influences the rate and the strength of contraction. The autonomic nervous system influences the rhythm, but there's really no need for external control. So if you've ever seen a movie where somebody rips somebody's heart out of somebody else's chest and they're holding it up and the heart is continuing to beat, this is something that would actually occur because the heart is autorhythmic. The stimulus to beat comes from within the heart itself and gets perpetuated across the heart. So the heart can continue to beat even if it's separated from the nervous system. This is different from skeletal muscle. Skeletal muscle wouldn't contract once it's separated from the neurons that control it. So here we're looking at cardiac muscle and some of these structures that I was just telling you about. You can see on the left there an illustration of cardiac muscle cells within the myocardium of the heart. You can see those striations, those bands of light and dark myofibrils. You can see the intercalated discs, those jagged lines that are binding adjacent cells together. You can see the Y branching structure between adjacent cells. And you can see those 1 or 2 centrally located nuclei that cardiac muscle cells will have. On the right, we can see into an individual cell. So you can still see those intercalated discs, those jagged lines. But now you can start to see some of the structures of the intercalated discs, including the gap junctions, those pores that allow messages to travel from the cytoplasm of one cell to the cytoplasm of another cell, and the desmosomes, those spot welds that are knitting those adjacent cells together. Also inside, you can see lots of mitochondria, and you can see those Y-shaped branches. Here's an actual view of cardiac muscle. So you can see the striations. Those are those lines that are going from the top of the screen to the bottom of the screen, the vertical lines within the cells. And then those dark lines that look like really dark striations, those really dark lines right here, those are the intercalated discs made of gap junctions and desmosomes. You can also see some Y branches. You can see the striations, and you can see the 1 or 2 centrally located nuclei within the cardiac muscle. These purple dots outside of the cells, those are some of the tissues that are surrounding the cardiac muscle. This is the endothelial layer that we'll get into when we get into the cardiovascular system. The last type of muscle is smooth muscle. Smooth muscle is found in the walls of the viscera, meaning hollow internal organs, things like the intestines, and within the airways, within blood vessels. And it's what allows for movement of the internal organs, movement of food through the digestive system, and it controls the walls of the passageways like blood vessels, the tracts of the respiratory system. It's present in the eye, where it's changing the size of the iris, adjusting the shape of the lens. And you all know that smooth muscle is present in the skin. There are tiny little smooth muscles that cause hairs to stand on end. That's that arrector pili muscle. So some of the traits that smooth muscle cells have, they're short fusiform cells. Fusiform means that they're shaped like a spindle, which is what Sleeping Beauty pricked her finger on, or shaped sort of like a football, meaning that they're wide in the middle and then they're tapered at each end. They have one centrally located nucleus. They don't have any striations visible under the microscope, although they do have myofibrils, and I'll show those in just a second. And these smooth muscles are generating slow, efficient, fatigue-resistant contractions, so they don't get tired. They can process your food all day long. And on the right there is shown some of the layers of smooth muscle that are surrounding the digestive system. And we'll talk more about those different layers when we get to the digestive system. So now, for now, just be familiar with the overall traits of smooth muscle. Here's what smooth muscle looks like underneath the microscope. So you can see those fusiform or football-shaped cells. You don't see any visible striations. You can see one centrally located nucleus. What you'll notice is it looks a lot like that dense, regular connective tissue that we saw in our histology unit, that dense, regular connective tissue that had sort of that wavy bacon appearance. Looks pretty similar to this smooth muscle. But in smooth muscle, the difference is that you're finding the nuclei within the cells. In dense connective tissue, the nuclei were outside of the fibers. Remember that in connective tissue, the fibers are in that extracellular space. And in smooth muscle, the nuclei are within the muscle fibers, within the cells. So I said that they don't have any visible striations, but they definitely have these myofilaments, actin and myosin, that slide past each other to allow for contraction. And it's organized in this more crisscross pattern throughout the cell. So on the left is a relaxed, smooth muscle cell. And you can see these triangular arrangements of thick and thin filaments. They're organized around dense bodies. These are just the anchoring proteins for these myofilaments. On the right, you can see a contracted smooth muscle cell, and you can see that the thick and thin filaments are sliding past each other. They're contracting the entire cell. So it's more like scrunching up the entire surface of the cell. And this is why we don't see visible striations in smooth muscle. You also won't see T-tubules or troponin. This is because most of the calcium that's needed to activate the smooth muscle concentration is coming from fluid around the cell. So we don't have that same triad organization within the smooth muscle. Finally, there are two types of smooth muscles. There is multi-unit smooth muscle, and there's single unit smooth muscle. And these two subtypes are basically organized according to whether they're connected with gap junctions or not. It's sort of a simplification, but we're going to go with that. So in the first type, multi-unit smooth muscle, This is where gap junctions are absent or rare, and cells act as independent entities. So in multi-unit, each cell is considered its own unit, and that's why it's multi-unit. This is present within the iris of the eye, within the walls of the blood vessels. All of the cells are acting independently of each other. In visceral or single unit smooth muscle, this is where gap junctions are really abundant between cells, and cells are acting as one single entity. So that's why it's called single unit. All of the cells are functioning as a single unit. They form sheets of muscle where the cells are connected by gap junctions. The muscle fibers can contract as a group because of these cytoplasmic connections. This results in rhythmic contractions, and these are found within the walls of most hollow organs, the viscera. That would be things like the intestines. So single unit smooth muscle, where there's lots of gap junctions, so cells contract at the same time, that's going to be functioning really similarly to cardiac muscle. Remember, those are all connected by gap junctions too. So cardiac muscle could be considered smooth or single unit, but we don't call it single unit, we just call it cardiac muscle. Skeletal muscle would essentially be multi-unit, right? Because individual fibers contract when they're stimulated by a neuron. One fiber doesn't affect another fiber. So multi-unit is found in the iris of the eye, walls of blood vessels, almost no connection between cells. Cells are acting as independent entities. And single unit found within the digestive system, anywhere that cells need to contract together. They have lots of gap junctions, and cells are acting as one single entity. And the last thing I want to talk about are some of the things that can happen to muscle. We talked about atrophy and hypertrophy. So in atrophy, if the muscles aren't used, you start to lose the bulk of the protein structure, and muscles get smaller and lose their power. You're not typically losing individual cells. There's not cell death. It's just removal of proteins from inside the cell. In hypertrophy, you're making more proteins and cells are expanding in size. If a muscle is strained, this is an overstretch of a muscle or a tendon. If you're getting a sprain, that's some damage to a ligament, some overstretch of a ligament. And if you've ever sprained a body part, you know that it takes a really long time to heal. Remember that ligaments are those dense, regular connective tissue. They have almost no vascularity, almost no blood flow, so it takes a long time to heal. If you've experienced muscle cramps or muscle spasms, this is something we don't really understand a lot about. The details are unclear with a lot of possible causes. Although we talked about one possible cause of muscle cramp, that is maybe myosin heads can't unattach from actin filaments in the absence of ATP. So one cause of cramping might be that muscles have contracted and they don't have enough ATP to unbind, to relax. We talked about myasthenia gravis, which is this muscle deficit that's due to autoimmune destruction of the receptors at that neuromuscular junction. So at the synapse, that presynaptic neuron is sending neurotransmitters into the synaptic cleft, but the postsynaptic cell, the muscle fiber, isn't getting the message because the receptors for that neurotransmitter are being destroyed by the immune system. Muscular dystrophy is an inherited muscle disorder, so it causes muscular degeneration in which abnormal genes interfere with the production of the proteins required to build muscle, and there are many types of muscular dystrophy. Multiple sclerosis is this, it's actually an immune system disorder of the nervous system that presents as a muscular disorder. And this is where there's an immune response to the central nervous system neurons that control the muscle fibers of the motor units. And this attack on the neurons causes chewing of this insulating material around them, the myelin sheath. So multiple sclerosis presents as a muscular disorder, but it's really the immune system attacking the neurons that control the muscular system. And we also talked about rigor mortis. One thing that you should know is that there's no muscle activity after death, but approximately 4 to 12 hours after death, the body becomes rigid, the muscles are fully contracted. So after death, there's some ATP remaining, there's calcium remaining, and calcium and ATP are the ingredients that are required for binding of myosin to actin. But after myosin binds to actin, the ATP that's required to unattach isn't there. So rigor mortis persists for a couple of days after death, and then rigor mortis ends when cells continue to decay and digestive enzymes are released from lysozymes, breaking apart those protein structures, and there's potentially digestion from microbes as well. So I hope this has been interesting to you. Make sure that you review all of Chapter 9 to prepare for the Quiz 5 that I'm going to deploy on Canvas on Monday. Please let me know if you have any questions about the material that I just covered or the material that I've been covering related to Chapter 9.